Okay, Assalamu alaikum, how are you everyone? I hope you are doing great in your foundation quizzes. Today, me, Adham, Amir, and Waleed and Asad are going to present for you the muscle chapters, the muscle chapter, of course. So inshallah, to be a quick revision, we will go through the most important concepts. If you have any question, don't hesitate to ask. Also, we'll solve some practice questions and know how to solve them and what should be your thinking process during solving such questions. So let's start. We are going to distribute it into three main parts. The introduction, which is the first 9.1 lesson, which is the muscle general functions and properties. It's general information. Then we are going to talk about the anatomy first, which is the structure of the muscles, and which will be mostly me. And then the muscle physiology. I will also be taking part of this, but mostly Waleed will cover this part. So let's start with the introduction, or the general. So general terminology. Before we start with any topic, especially in medicine, before you start with any topic, studying any topic, you should know the general terminology of this topic, because there are certain letters or words which are going to be like repeated because they have the meaning of the main topic you are going to study. For example, myo or mes both mean muscles. So because we are studying muscles mainly in this chapter, you are going to see that they are being repeated in many words or scientific words. Sarco also means flesh, which will also be referring to the muscles. So we are going to see. We have two examples, which are the sarcolemma. Sarcolemma is the plasma membrane of the muscle cells. So it's just plasma membrane, but a special name for it to indicate that it's for the muscle cells, not any other cell. Also the sarcoplasm, which is the cytoplasm of the muscle cell, as you can see here. This is a muscle fiber, which is the muscle cells. We are going to explain it now in details. As you can see, the plasma membrane is called sarcolemma, and the cytoplasm, which is the fluid inside, is called sarcoplasm. Okay? So, you know, of course, we have different types of muscles. We have skeletal muscles, cardiac muscles, and smooth muscles. We will be focusing mainly on the skeletal muscles in this chapter, but you need to know some keywords that indicate if they are asking about the cardiac muscles or the smooth muscles. So, for example, at the beginning, skeletal muscle, from its name, it indicates what? What keywords can you think of when I tell you skeletal muscle?
That is attached to bones.
Okay, attached to bones, and I hear the voluntary. Okay, any other keywords? Exactly. So skeletal means it's actually attached to bones, which causes movement, as we're going to see. Striated, which means there are black and white stripes. You can remember it like zebras, for example. We're going also to see what is the reason behind this striation. And they are voluntary, which means they are under your control. If I want to hold my phone, I'm going to pick it up by controlling my hand muscles, for example, or my arm muscle.
Sorry.
So what about the cardiac muscles? What can you think of? Okay.
It's involuntary.
Okay, involuntary, so you can't control it. Any other? Okay, it's also striated. Okay.
Intercalated.
Exactly. So also one of the keywords is intercalated discs. They are connected by intercalated discs. And cardiac means, okay, so cardiac means it's in the heart. So this is a special type of muscle. It's only found in the heart. Striated, it's striated like the skeletal muscle. So this one is not special characteristic because it's shared between both. And involuntary. If I told you to increase your to increase your heart rate right now, or stop it for 5 seconds, you wouldn't be able to do that. So you can't control it. Okay, and for the last one, which is the smooth muscle. Okay.
It's in hollow organs and it's involuntary.
Exactly. Any other from its name, smooth?
And there's no structure.
Elongated.
Okay. So yeah, it's in visceral organs such as stomach, urinary bladder, internal organs. It lines the internal organs, which needs muscle. It's smooth, non-striated, so you can see these black and white stripes, or dark and light stripes. And it's also involuntary, like you can't control your intestinal muscles, for example. Okay, so let's start with talking about the general characteristics of muscle tissue, especially the skeletal muscles, because as you said, we are going to be our main focus is on skeletal muscles. So, what characteristics of skeletal of muscle tissues in general? Especially, okay, elasticity.
They can contract.
Okay, contractility, exactly.
Excitability.
Okay, and
And responsive.
Yes, responsive. Okay, so excitability or responsiveness means that it gets activated or it reacts to external stimuli, as we are going to see in the physiology part, which will be the action potential. Contractility means that it can change its length. Actually, so the contractility means it can change its length, which causes the body movement. Most of the, or all of the body movement are by the contractility of the muscles. Extensibility, which means that it's not only contracting, or the length is not only decreasing, it can also stretch beyond its normal length. So this is extensibility. And don't get confused between extensibility and elasticity. Extensibility is from extension, and it's being increased in length. While elasticity is the ability to return back to its original length after this stretch is gone. So these are the four main characteristics of muscle tissues. They are easy, but they're still very important. You have to know them. So here we have a simple question. Who wants to? Okay, so I'll read it first. While investigating the role of muscles in the body, you find a tissue that can extend or stretch even beyond its resting length. What is the term for this describe? What is the term that describes this characteristic? Okay. Exactly, extensibility. So don't confuse with elasticity, because they may seem at the beginning confusing. So yeah, it's extensibility. So also this one is very easy. What are the functions of muscles in general in our body? Okay. Sorry? Exactly, produce movement. What else? Okay.
Heat, insulate heat.
Insulation, it's mainly for the fat or adipose tissue, but okay. It produces heat, it doesn't insulate. Okay.
For the posture.
Exactly, maintaining posture. Any other? Okay.
Also stabilizes our joints.
Exactly. So the same. It can be the same as maintaining posture, but so producing movement. First of all, course, all the body movements are due to muscle contractions, as we're going to see. Maintaining posture and body position. Like right now, I'm standing straight because of muscles. Okay, and also actually, we're going to explain the muscle tone, which is that the muscles are a little bit in a contraction state all the time, even if you are not using it, which is causing this posture to be maintained, stabilizing joints while movement. Okay, and also generating heat. How can we generate heat using muscles? Exactly, shivering. Okay, so nerve and blood supply. Of course, you know that all cells in the body needs to be all of the body cells need to have blood supply, so the oxygen reaches, and also nerves, so the brain can control it and you can control it. So first of all, generally, each muscle has at least one of everything: one nerve, one artery, and it can have one or more veins. So at least one, and for the veins, it can have more than one, because the veins usually tends to be smaller. So sometimes you need more than one. And muscles have high blood supplies. Why do you think this? Why do you think they need high blood supply? Okay.
For oxygen and nutrients.
Okay, but all cells need oxygen and nutrients. But the muscle cells have exceptionally high blood supply.
To use them to generate energy for contraction.
Okay, so you need to.
Because they need to use O2 and nutrients to contract, and they need ATP.
Exactly. Okay. So in the normal state, like right now, we are just sitting. You don't need any muscles are not supplied by with the maximum capacity of blood they can get, because they are not really using them a lot. But during intensive or high exercise, muscles can take up to 80% of the whole body's blood supply. Because they need a lot of energy, as we are going to see in the physiology. They need a lot of oxygen and a lot of nutrients to convert it to energy, as you just said. So that's why it needs a lot of blood. Okay, so let's take it from the outside in. Of course, the muscles should be covered to be protected. So they are covered with connective tissue, sheath, and they are covered with multiple ones to ensure that they are properly protected. Because also they are highly movable organs, so they need to have protection. So we have three types of connective tissue sheath which are surrounding the muscles. From the outside, we have epimysium. Epi means around, so it's around the whole muscles. You can the whole muscle. You can remember it like this: epimysium. So epi means around. Peri is around fascicles. So we are going to ask what are fascicles. We are going to see it in the next slide. Fascicles are group of muscle fibers. And what is muscle fiber? Muscle fiber is the cell. So a single muscle fiber is, you know, the cell. We used to draw a circle and then the nucleus and some organelles. But for the muscles, it's different. It's not like circular shape, it's cylindrical shape. We're going to see it now with characteristics of muscle cells. So perimysium is around fascicles, and fascicle is just a group of muscle cells or muscle fibers. And endomysium, which is the innermost layer around each single muscle fiber or each single muscle cell. They are both the same thing. Also, I found this info in the book. It's not any. It's good to know. Okay. You have a question?
I think the endomysium surround the sarcolemma, not the
Sarcolemma, as just said, is okay. I will go through it fast. Sarcolemma is a plasma membrane, which is the outer layer of the muscle fiber. So yeah, you're correct. It's surrounding the muscle fiber from the sarcolemma. Okay. Okay, so epimysium and perimysium are both dense irregular connective tissue. You just have to know it. While endomysium is areolar connective tissue. I guess you should know by now what is meant by dense irregular and areolar from the first chapter. Okay, so here in the picture to easily identify them or remember them, the epimysium is around the whole muscle. Here we have each one of these is muscle fiber, okay, or a muscle cell. Okay, so each one of these is muscle fiber. A group of them will be surrounded by perimysium, and each individual one will be surrounded by endomysium. Before we continue, that question till now? Okay, this part is easy. So muscle attachment. As we just said, muscles aren't floating freely. They are attached to bones, especially the skeletal muscles which we are discussing. So we have 2 points of attachment for a muscle. One is called the origin, and one is an insertion. Okay, so the first one, it is more proximal to the body. Do you know the meaning of proximal? Do you what? Sorry? Exactly. So it's closer to the body. Okay, and it's not as movable. So it's not movable at all, or little mobility. It depends on the muscle. While the insertion is the more distant, so it's further away from the body compared to the origin, and this is the movable part. Let's take an example of this. So we're going to see an example also after this one. So let's say this is a body, and for example, my biceps. We have here 2 attachments. This one is the origin, I'm going to show it here, and this one is the insertion. So when I do like this, which one is the moving part? Insertion is moving toward the origin, but the origin, if you point it out, it's stable in its place. So as you can see, in relaxed state, origin and insertion are in their normal position. When the muscle is contracting, the insertion will get closer to the origin, which is closer to the body. For example, here, as you can see, the origin is stable in its place, and the insertion is moving toward it, and it's more distant. Okay, so also we have different types of attachment, which is the direct fleshy attachment or indirect attachment. From its name, what do you think it indicates?
Attached to the bone or the cartilage direct.
Exactly. So the direct one, it's directly attached to the bone itself. But okay. So yeah, it's directly attached to the bone. But like the muscle exactly, bones and cartilage also have an outer layer. For the bone, it's called periosteum, and for the cartilage, it's perichondrium. So it's the exact same idea of epimysium, for example, but for bone or cartilage. So when they are directly attached without any special structure in the middle, it's direct attachment. But the connective tissue forming the epimysium can form special structure in between, such as a tendon, if it's rope-like, or a sheet-like aponeurosis. So you have to know them. If it's rope-like structure, it will be called a tendon, and if it's sheet-like, it will be called aponeurosis, and it will be between the epimysium and the periosteum. It will be connecting both of them together. Okay. So which one do you think is better? Which one is better in your opinion?
The indirect is stronger.
Okay, so both of them, of course, are different types. Of course, have different abilities. So both of them are important. But yes, indirect attachment are much more common in the body. because they are stronger and also they protect the muscle itself from the sharp edges of bones. When there is a little bit space between the muscle itself and the bone, it's less likely to get scratched by the bone itself. Okay? And you have to know the tendons are mostly tough collagen fibers which are very strong. They are actually stronger than steel fibers of the same width. So yeah, that's it. It's highly strong. Okay, here's a question for you guys. Which of the following is an example of the direct attachment for muscle? We just mentioned it. Okay. Three.
No,
Okay.
no, it's 4.
4, epimysium of muscle attached to bone.
Yeah.
Yes, exactly. Why? Because it is directly attached. Okay, so okay, thank you. Okay. Another question. During a surgical procedure involving muscle repair, which connective tissue would the surgeon typically encounter to cut a muscle fascicle open? Okay. Perimetrium, yes, exactly. This one is easy, but don't get confused of periosteum, which said this one is for the bone, okay? And also the key word here is fascicle, which is group of muscle fibers, so that's why we chose perimetrium. Okay, now we are done with the outer covering or the connective tissue sheath of the muscles. Now let's study the muscle itself now. So you know muscles are organs, of course. So the whole muscle, which is covered by epimysium, is made from even smaller structures which are called fascicles. We also mentioned them, which are groups of muscle fibers. Now let's concentrate on the muscle fiber itself, because as we said, they are cells, but they don't seem like the typical cells we imagine when someone says cell. So what are special about muscle fibers? What makes them special? Special characteristics of muscle cells?
They're cylindrical.
Okay, cylindrical in shape. Any other? Striated, okay.
Voluntary.
Okay.
Stimulated.
Stimulated?
They get stimulated.
Okay, so yeah, exactly. So
لا
first of all, it's cylindrical, as just said. It's longer than normal tissues, like it can be very long compared to other tissues. It's also multinucleated, like a single muscle fiber can have up to 1,000 nucleus. But our ordinary cell only have one nucleus. So it's multinucleated, it's cylindrical in shape, and it has some special structures and organelles that are not found in any other cells. Let's start with the first one, which is myofibril. Myofibril is actually an organelle, like the mitochondria or nucleus, but it's only found in muscle fibers. And actually, myofibril takes up to 80% of the muscle fiber space, so it's very dominant here. And these are the reasons for two things. First of all, is the striation, as we're going to see now, and the other one is being able to contract, because they have the functional unit we're going to talk about now. So let's take the myofibril. Myofibril is made even of smaller units, which are called sarcomere. So are you with me? You are getting it from the bigger to smaller. Muscle, fascicle, muscle fiber, myofibril, and then sarcomere. Okay? So sarcomere is the functional unit of the muscle. It's the contractile unit. So a single sarcomere can contract on its own, or form the action that will lead to contraction. But of course, we need millions of these, or even billions, for a muscle to contract, because they are very small structure, microscopic structure. So let's continue. Here's the myofibril. Myofibrils, we said this is a myofibril. So let's break down the myofibril into its smaller sarcomere. As you can see, even the sarcomere have different parts or different names for different parts. First of all, you need to know it's mainly composed of two proteins: actin filament, or the thin filament, and myosin filament, which is the thicker one. Okay? Second. Okay. So actin filament is the blue thinner one, while the myosin filament is the thicker red one, as you can see here. So how does these two appear in different parts? So they name the parts according to the proteins that are available or not available in the part. So any part where the myosin is available or appearing, they call it A band. Okay, and the A band is usually thicker and darker than the I band. Why? Because it has the thicker myosin filament. So any place where there is myosin, it will be called A band, even if actin is overlapping or not. So this part is A band, this part is A band, because it is determined by the myosin filament. While the I band, which is the lighter portion of the muscle cell, how do you determine this part is I band? Because it's only made from actin filament. It doesn't have myosin. So only this part, or no, this is half of it. This part, because it doesn't have the red thick filament, is called the I band, which is the lighter one, lighter in color. Okay. We have also specific zones. For example, here you can see this zone. It's part of the A band, correct? Because it is from the myosin filament, but it doesn't have actin. So we will call it H zone, which is lighter. So the A band is determined in all of the areas that have myosin, okay? And we have two parts. One where actin is overlapping, you can see both of them above each other, and one where the actin is not overlapping, there is no actin here. This part is A band, okay, and it doesn't have a special name, but the part where there is no actin, it's called H zone, which is also lighter, a bit lighter than this because it only has the thick filaments, not both of them, okay? In the middle, we have M line, which is made from accessory proteins, mainly from myosin. It's very important to know it. I wrote it, we'll see it now. So the M line is made from accessory proteins that is attached to the myosin filaments to stabilize them and keep them in place. Okay, and the Z disc is actually between the Z disc is the borders of a sarcomere. So from where exactly do you start and say from here to here is a sarcomere? It's by the Z discs. It's also made of a couple other proteins. We are going to see them. But mainly we have the start of the elastic filament. Okay, here what I just said, the H zone, M line, Z discs. One second. Okay, so before we continue, keep in mind also that sarcoplasm, which is a cytoplasm of the muscle fibers, is rich in glycosomes and myoglobin. Glycosomes, of course, are storage granules for glycogen, and myoglobin is like hemoglobin but inside the muscle. Why do you think they need glycosomes and myoglobin? Why are they important to
store oxygen?
Okay, oxygen. Okay.
For ATP.
Okay, exactly. So as we said, muscle needs a lot of energy, so it needs glycogen to turn into glucose and then to ATP, like we're going to explain now, and also oxygen. Okay. It's very important to know if we cut at each part which we just mentioned, what are we going to see? So at the I band, you cut in the I band and like check what is this, or what do we have in this part. You will only find I band. You will only find thin filament, which is actin filament. Because this is the definition also of I band, right? It's the part where there is no myosin, it's only actin, which is the thin filament, okay? But if you, for example, cut in the middle of an A band, you will see both myosin and actin. And actually, they discovered that there is a special arrangement for these filaments, so they are not they are arranged in a certain way where each one myosin filament is surrounded by 6 actin filaments, while each actin filament is surrounded by only 3 myosin filaments. So are you with me at this point? So you can see they are forming a hexagonal shape. The thin filaments are forming a hexagonal shape around the thick filament. Okay? Here, if you cut in the M line exactly, you'll find 2 things, as you said, the myosin itself, which is the thick filament, and the accessory proteins, mainly the myosin, which forms the M line. And at the H zone, we find only thick filament, okay? Are you confused about this part, or is it okay? Okay. So, a question, in a cellular anatomy study, you encounter a segment of a myofibril that is described as the contractile unit. What is this segment called? Okay. Yes?
Sarcomere.
Sarcomere. So as we just said, sarcomere is the contractile unit of the muscle, okay? Okay. So sarcomere is even made of smaller structures which are called myofilaments. And don't get confused between it and myofibrils because at the beginning I was confused about them. So you can remember it, myofibrils comes from fibers, so they are still the bigger part. But myofilaments are much smaller, okay? Which are mainly the myosin and actin. You are going to know the structure in details of each one. Not in details, but in some details about them. So first one is the myosin, which is thick. It is usually red, or it's red in this picture, and it's made from myosin molecules. We're going to see that now. This is just a recap of what you just said. Okay, so we have thick filament and thin filament. Thick filament is the myosin. What is the myosin made from? First of all, it has two parts: the tail, as you can see, and the head. The head is the actin binding site. We're going to see now in the physiology part, or how the muscles work. How does a muscle contract? It is just by moving the actin, or sliding the actin along this vertical axis. Horizontal axis like this. So it's just like sliding above each other. So the myosin will just attach itself to the actin and then push it towards the midline. So we need an active site. This is considered a reaction. So there is an active site where the reaction between both of them is going to happen, and we can explain now in details. So here's the binding site. The thick filament is made out of 6 polypeptide chains. So how do you remember? How can you remember the number easily? Because we have 6 thin filaments surrounding each thick one, and the thick filament is made out of 6 polypeptide chains. So you can remember it in this way because they have the same number. How can we distribute the 6? 4 light and 2 heavy. The 4 light, as you can see, each one is made of 2 strands twisting around each other. So each strand is made of 2 light. So we have 2 lights from this one, 2 lights from this one, twisting around each other, forming the tail. Okay. The head is an extension of the tail, so it also has the light chains. But at the active site, you have the 2 heavy chains, one in this head and one in this head. So is it clear? The light chains in the tail and the heavy chains in the active site. Okay? Everything is okay? Okay, let's continue. So it's written here that 300 myosin molecules make a single myosin filament. So this is the whole filament. As you can see, it has a lot of molecules. And this is one molecule. 300 of these together will make one filament, which we just saw in the picture. Okay. So now let's talk about actin. Actin is also associated with different proteins, with other different proteins. So let's start with actin. First of all, actin is made from globular proteins, which you can see they are like beads or kidney shaped, as you can see. So G-actin is a single bead, like this one. When they are joined together to make this like strand shape, they are called F-actin, which stands for filamentous actin. Okay, filamentous is a filament. So this is the building unit. It forms this one. Okay. And of course, we have the actin myosin binding site also on the actin or globular protein, which will join together to make the muscle contract. Okay. Actually, actin is also like there are many regulatory proteins associated to actin, so we need to know them as well. First one is tropomyosin. As you can see in this picture, actin isn't alone. There is this yellow or orange thin strand that's covering all the active sites, as you can see. So what is tropomyosin? Sorry, what is tropomyosin? It is protein that is covering all the active sites of the actin to prevent contraction until the signal reaches or until the correct timing is here. So I like to make an example of bed sheet. It's covering the mattress, for example. When you want to reveal a mattress, you just pull the bed sheet and everything beneath it will be released. So it's the exact same thing. It's just a cover to the binding site. For the troponin, I like to call it the glue bead. Why? Because it's also globular protein. It's as you can see, it's also bead shape. But why glue? Because they join everything together. First of all, we have 3 binding sites which enables it to join everything together. The first one attaches the troponin itself to the actin because it needs to stay there, right, to regulate the tropomyosin, as we are going to see. The second subunit binds tropomyosin. So from one subunit you are binding the actin itself. From one subunit you are binding tropomyosin. So you are joining them together, correct? And the third one is for calcium ions, which is the signal to start contraction. We are going to see that now also. And the elastic filament, I mentioned them, made from giant proteins called titin. This, it's not, a second. Yeah, this yellow protein is the elastic filament. It's made from titin. You have to know it's made from titin. It's very important. And it seems that it ends here, but actually it extends from the Z disc, as you can see, and it goes through the myosin filament until it reaches the M line. So it actually connects the M line with the Z disc through the myosin filament. Okay. Okay, some other proteins where the function was not mentioned clearly for most of them. Dystrophin, no, this is a mistake, it's not bitchy. So dystrophin links the thin filament, we need to see the picture here, to the integral protein receptor. 1 second. Okay, so we have a protein which is called dystrophin, sorry. This one, it's also not showing this picture, but you have to imagine it. It's connecting each thin filament to integral proteins in the sarcolemma. What is a sarcolemma? Just mentioned. Exactly. So we have a protein that connects each one of this to the plasma membrane of the muscle fiber, which is very important. We're going to see now in maintaining the integrity of the sarcolemma itself and of course the thin filament. And the more connection you have, the stronger structure you have. That's why we have all of these proteins connected to each other. Another one is, okay, these just have to know that they are there for binding. We didn't mention any special characteristics for them. Nebulin, myosin, which is actually also found in the M line, and C proteins. Okay. Last protein finally is the dismal, intermediate dismal. It extends from the Z discs and connects different myofibrils together. So this one may seem similar to this one, but it's not. Let's compare them. So the dismal joins different myofibrils. Like, you get me? 1 second. Myofibrils, we said there are many of them in one muscle fiber. So it's connecting the different myofibrils together. While the dystrophin is connecting the actin to the sarcolemma of the same myofibril. Okay? So this one is in the same myofibril, and this one connects different myofibrils together. Okay, we have a question here. Which proteins, if following, are seen in a cross section at the M line? Okay. Yes.
Myosin and myosin.
Exactly. So, a very useful way to solve question if you don't remember on the spot is actually elimination. So, of course, you know at the M line we don't have any actin, so we'll cross these. And, of course, if you remember it, it's easier to solve, of course. So, okay. Which of the following is a function of dystrophin in the muscle cell? Okay, it attaches a thin filament to the sarcolemma. So the dystrophin is in the same myofibril, okay? As you should know by now, homeostatic imbalances are very important to know and focus on. So the first one in this chapter is describing the muscular dystrophy. Muscular dystrophy is a general term which indicates a group of genetically inherited diseases that affect the muscle and cause them to be weaker and degenerate over time. They are very dangerous actually because the affected muscles, such as the cardiac muscle or chest muscle, they can prevent blood pumping or bleeding, which is fatal. Usually it appears during childhood between 2 to 7 years old. And at the beginning, actually the muscles seem to grow, not to decrease in size, although the muscle is decreasing. Why? Because it's accumulating fat and connective tissue, while the muscle fibers itself are getting degenerated and decreasing in size, which is causing the weakening of the muscles. So here we have a more specific example mentioned in the book, which is Duchenne muscular dystrophy or DMD. This one, this disease is caused because a genetic defect in the dystrophin protein. So the muscles of the children who have DMD doesn't have dystrophin. Do you remember what was the function of this protein? Just mention.
Binding actin to saccharoline.
Exactly. So why is it a big deal if this protein isn't there? Because actually it's increasing, as we said, the structural integrity of the sarcolemma itself. So when you don't have this protein, the sarcolemma will tear from contraction. So what will happen when the sarcolemma tears? The extracellular fluid will join. Exactly. The extracellular fluid will join with the cell internal structure, right? Because it doesn't have plasma membrane anymore, which will cause a lot of extracellular calcium. Exactly. We are going to see why calcium is our main concern in the physiology part. Will damage the contractile unit, which is sarcomere, and will destroy its regenerative capacity. So the muscle without sarcolemma won't even have the ability to repair itself. So it will be damaged forever, and it will even keep getting worse. Okay? Actually, there is no treatment for this disease. The only thing is physiotherapy, by trying to keep these children mobile as much as possible to prevent joint deformities and to delay muscular weaknesses. Okay, we have special. There are some special structures in the muscle fibers. Sarcomere we already discussed. Now we are going to discuss the sarcoplasmic reticulum and T-tubules. For the sarcoplasmic reticulum, it's a special type of endoplasmic reticulum. So, and it's only found in muscles as well. So it's especially smooth endoplasmic reticulum. Its main function in muscle is to store calcium, store and control the movement of calcium. We're going to see this now in detail. And where are they? They are. The picture is easier to. So this is dark blue structure. This is the sarcoplasmic reticulum. So they form like a web around the M line, okay, and they form here it's like the end because it's separated with another structure. We're going to see it now. So here it's called the terminal system. So terminal system is the end of this sac structure of the sarcoplasmic reticulum, and here we have a web-like structure in the M line, okay? And this whole structure, the blue one, is called sarcoplasmic reticulum. It's storing calcium, okay? We have in the middle what's called T-tubule as well. And where can we find the T-tubules? On the Z discs, as you can see. And what are T-tubules? They are protrusions of the sarcolemma deep inside the cell or the muscle to reach each and every single myofibril, as you can see. So, if you have a cylinder, for example, and you have cells on the periphery and cells in the middle, if you don't have any structure to send the signals, which are mostly outside, to the cells inside, they won't participate in the contraction, right? So we need these structures to make sure that every signal reach to the deepest part of the muscle and to reach myofibril to contract. So the T-tubules is at each A band, I band, A band, I band, that change from this one to this one is the junction between the both of them. Okay. It's okay. We said most of this. Okay. So what are triads? Triads are just like grouping thing. The structure that is formed by two terminal systems, and the T-tubules in the middle is called a triad. Yeah, so T tubules, two terminal system on each side is a triad. Okay, so now we're going to start digging even deeper in the muscle contraction, the sliding filament model of contraction. Like, how does muscle contract? What causes its length to decrease and then return back to its normal length? So first of all, there is important concept that you should be familiar with, that it's not actually decreasing in length. Any of the filament, the actin filament, if it's, for example, 5 micrometers, it will always be 5 micrometers. But if it's sliding above the myosin filament, it will make the whole muscle decrease in length. But the filament itself doesn't change in length. Okay. Okay. So this part is easy, but it's still very easy to make mistakes in. I personally made one mistake in this part, although it's easy. So what are the changes that you can see in a contracted muscle? What are the changes you can see in a contracted muscle when you compare it to a normal muscle? So here is the normal formation we just described, the H zone, the Z disc, and everything. When the muscle contracts, there are some or several changes happen. As we said, the filament itself doesn't change its length, but the A band shortens. It doesn't disappear, but it shortens. The I band, sorry, I said A band. The I band shortens. Why? Because the actin filament is moving closer to the M line, right? So the segment with only actin filament is decreasing. The distance between two Z discs will decrease as well. H zone will disappear completely. What was the H zone?
It's just the myosin on its own.
Exactly, in the middle when myosin isn't overlapped by any actin. And the A band, they move closer to each other. Like A band 1 and A band 2, they will become closer to each other because the I band decreased in length. While the A band itself won't change in length. Why? Because the myosin filament isn't moving at all, isn't sliding. Only the head is moving to moves the actin filament. You got this part? So the A band itself doesn't move. That's why it doesn't change in length. Which will cause the A band to the thick filament, which will cause the A band to have the same length. So as you can see here, the A band is the same. H zone is completely disappearing because the actin filament came here. And the Z disc between this and this is much longer distance when compared to Z discs in a contracted muscle. Okay, so now my friend Waleed will continue with you the muscle physiology, starting with muscle contraction.
Okay, assalamu alaikum, hello everyone. I will start now with 9.4 about muscle contraction. This is going to be exactly about physiology, so please try to focus and concentrate with me. And if you miss anything, please tell me so you can follow up and keep it. Okay, so I'll start with a small recall and some information that you already know and my colleague already explained. So first of all, skeletal muscle contraction are voluntary. So skeletal muscles, they are voluntary. They contract in response to motor neurons that are in the brain and spinal cords. However, the actual contraction of the skeletal muscle, it involves only few motor neurons that are in the spinal cord. Motor neurons are the connection between the nervous system and the skeletal muscle. So it transports the command to contract from the skeletal nervous system to the skeletal muscles. And as my colleague already explained, neurons and muscle cells, they are both excitable. What does excitable mean? Does anyone know? Yeah. Yeah, by changing their membrane potential. Okay, so neurons and muscle cells, they change their membrane potential in response to stimuli. Okay, so how does they really change their membrane potential? This principle depends on the ion channels. So ion channels, their opening and closure play a role in the changing of membrane potential. And we have two types of ion channels that are important for excitation and contraction of skeletal muscle. We have the chemically gated and the voltage gated ion channels. For the first type, chemically gated, as it explained itself, it's chemically gated. So it's open and closed in response to chemicals such as neurotransmitters and acetylcholine. And they cause a small change in the membrane potential called graded potential. Okay, for the voltage gated, they open and close in response to ion change, and for the graded potential that's produced by chemically gated. So chemically gated, they open in response in response to chemicals. They produce a graded potential. This graded potential will affect the voltage gated and cause the production of an actual potential. So for the motor neurons and neuromuscular junction anatomy, we have here a motor neuron, as you can see. So it's cell body. As you are going to study later in the nervous system, the neuron muscle of the neurons, they consist of cell body and appendages and the extensions called axon. So their cell bodies are the spinal cord, and they produce an extension that go into the muscle that they want to innervate. So that's called an axon. So since the axon, when it's entered the muscle, it's going to branch into axon terminals, and each one will supply a different muscle fibers. So one axon at the same time can supply many different muscle fibers with its axon terminals. Okay, so for the neuromuscular junction, as it explains itself, is the junction between the neuron and the muscle. So it consists of three parts: the neuron part, the muscle part, and the junction of the space between them. So for the neural part, as I already explained, it starts with the axon the axon terminal. Axon terminal contain calcium channels and also vesicles that contain the chemical that's going to be released and going to do all that. So we have also the muscle part, which is the sarcolemma. As you can see here, we have the sarcolemma. There is junctional folds. Does anyone know what's the function of the junctional folds? Yeah, it increases the surface area, sure. So in this area, small area, we have thousands of receptors, of acetylcholine receptors. And the space between them is called the synaptic cleft, is filled with extracellular substance, such as it's rich in glycoproteins and also collagen fibers. Is it okay? Okay, can anyone answer? Okay, after acetylcholine binds to its receptor on skeletal muscle cell, which of the following events is triggered? Activation of a G protein pathway indirectly affecting calcium levels and modulating contraction, or opening of chemically gated channels which lead to localized depolarization, closure of sodium channels leading to membrane regularization, or direct contraction of the muscle upon acetylcholine binding. Yeah. Second one, right. So I'm going to explain it. Sorry. So I will start now with the real physiology and the real contraction of the muscle. So it's divided into 4 stages. The first stage is which is the events at the neuromuscular junction. As I just explained the neuromuscular junction anatomy, I will explain the physiology and what's really happening here. So the action potential will be transported from the cell body into the axon through the axon, and it will arrive here in the axon terminal. The axon terminal, the action potential will cause the open of calcium channels. So what type of channels they are? They open in response to action potential or membrane potential. So are they chemical or voltage gated? Voltage gated, yeah. Okay, so the calcium will enter, and this enter of the calcium will cause the release of the vesicles, which contain what type of chemical? It contains chemicals such as acetylcholine. The acetylcholine then will go through the synaptic cleft, and it will bind to with the acetylcholine receptors on the sarcolemma of the folds, as I said. Okay, so this binding will cause the open of the channels, the acetylcholine receptor channels, which are chemical gated, and will cause the sodium to come in and potassium to come out. Okay, so if the sodium is coming in and the potassium is coming out, what's the difference? There will be nothing, right? No. Yeah? What's going to be?
It will depolarize, it will get
Yeah.
less negative.
But I mean, if we have the same amount of sodium that's coming in and potassium that's coming out, will be there any difference?
No.
No. So we have more sodium that's coming in than potassium that's coming out. For that reason, yeah?
3 sodium in, 2
Yeah, 3 sodium in and 2 potassium out. For that reason, there will be more positive charge that will happen in the middle. Okay, so this local depolarization, and then, okay, so after that, is the acetylcholine will be always, okay, so it's permanent spikes and we will always be contraction? That's not the case in real life, right? So we need to terminate or end its effect by removing it through the acetylcholine esterase. It's an enzyme that is responsible for destroying the acetylcholine after it binds and the action potential is already finished. So we have this homeostatic imbalance. It's very important. Each homeostatic imbalance you should memorize everyone, and many questions in the exam they are coming from this. So In this hemostatic imbalance, talking about the conditions or the problems that we usually have at the neuromuscular junction. Okay, for this example, there are many toxins, drugs, and even diseases such as myasthenia gravis that interfere with the acetylcholine receptors. For that reason, if we don't have any acetylcholine receptors, there will be no graded depolarization, there will be no action potential or no contraction at all. So we will have no contraction. Its symptoms include dropping upper eyelids, difficulty swallowing, and also talking and generalized muscle weakness. And myasthenia gravis as an autoimmune disease. So an autoimmune disease, it means that the body itself is attacking its own cells and own receptors. So the body is attacking their own receptors of acetylcholine. So there will be no contraction and will be all of these symptoms. Okay. So for the second point, we have the generation of an action potential across the sarcomere. So we already reached the graded potential, or as it's called also, the end plate potential. It's the graded potential that's produced by acetylcholine binding the sodium in and the potassium out. This is called the end plate potential, a graded potential. And this will cause the opening of the voltage-gated sodium channels. This will produce the reaction potential, and it will be transported through the sarcolemma until it finds the T tubules. Okay, so we produce the action potential. Then will be a period of refractory period or called repolarization, where we are opening the calcium channels after we finish contraction. So the voltage potassium channels will lead to the go out of the potassium, and this will cause the repolarization. Okay, so depolarization, it will become more positive since the sodium will come in. Repolarization, it will become more negative since the potassium is going out. Okay, for during repolarization, we'll have a refractory period where the muscle cannot be contracted or stimulated anymore. So it's going to reset itself to the rest state and imbalance. It will restore its imbalance and its normal condition, and then will be contracted again. Okay, as you can see here, there's the rest membrane potential. It's -90. Then we have the graded potential here right now. It will cause the opening of the sodium voltage-gated channels, will reduce the depolarization of the action potential. Then we have the repolarization due to the potassium is going out, so the positive charge will be less. And after that, we have the closure of the channel and this refractory period until it becomes to its normal resting membrane potential, and it will continue like that contracting. Okay, can anyone answer?
It's basically into potassium out.
Is it possible? Is it got?
Oh, then it's opposite.
Yeah.
It's opposite.
So what's the answer?
So 3 out,
3 sodium out and 2 potassium in. So here we are going to talk about a different object that I'm going to explain right now. We're talking about the sodium-potassium bond, ATPase bond. It's not the one you already saw. That's right. So for excitation contraction coupling, as you can see here, the action potential will be transported through the sarcolemma. It will it will be at the tubule, as you can see, it will go down. And this going down, the action potential will, so this will cause the change of voltage-sensitive tubular protein. This part is very important. Please focus with me, since there was last year a very important question about this, and everyone got it wrong. So what's really happening here is that the voltage-sensitive tubular protein, not the calcium channel, the voltage-sensitive proteins will undergo a conformational change. They will change their shape. And this change will cause the opening of calcium release channel. So the calcium releasing channel is not really voltage sensitive. It's open in response to the change of the potassium, the proteins. So the proteins itself, they are voltage sensitive. And the calcium will be released from the sarcoplasmic reticulum and will enter the cytoplasm of the muscles.
So the proteins are voltage sensitive?
Yeah. The proteins are voltage sensitive, while the calcium channels, they open in response to the change of the protein itself. They are not opening to the action potential. Okay, so for the excitation contraction, so we have a problem here. I already added a question that we had last year, and it was removed, so I will just explain. So the question was, which of these events will contribute to the muscle contraction or the excitation contraction coupling? So the choices was the voltage-sensitive calcium channels, opening of the voltage channels, or opening of the calcium channels, or opening of the chemically gated channels. The right answer was the calcium channels. They are not voltage, so that's wrong since they are opening to the mechanical change in the proteins. So you got my idea?
What was the question? What was the question?
ما في مجال أهم السؤال. Okay. So I will repeat the question. So the question was, which of the following events will be contributing to the excitation contraction coupling? The choices was the opening of the voltage channels, the voltage dependent, or the chemical dependent, or the opening of calcium channels. So you would say that all of the three answers are correct. However, the only correct answer is the calcium channels, since the calcium channels, they are mechanically, so they do not open in response to chemicals or even voltage. So as I explained now, they open in response to the change that's happening in the protein, not to any chemical, not to the action potential.
So the protein is what, like the change in the protein is what it's reacting to?
Yeah, so the protein itself is sensitive to the action potential, voltage sensitive. While the calcium channel, calcium release channel, it's not triggered by action potential. It's triggered by this information channel. I wish you get my idea. So that's it. Then the calcium will be going into the cytoplasm of the muscle cell. It will bind to the troponin. As my colleague explained, the troponin, it binds to the C part of the troponin. And this will lead to the movement of the tropomyosin that was blocking the myosin binding site on the actin. So, just as my friend said, you are removing, for example, like a bit, you are removing the bit. Yeah. And the myosin binding site on the actin will be released, and the myosin will bind to the actin. So now with the cross-bridge cycle, the myosin, the cross-bridge usually starts with the binding of the myosin to the actin. The myosin at the start of the cross-bridge will be at the highest energy state. It will be binded with ADP and inorganic phosphate. The next step is the power stroke of the myosin. After it binds to the actin binding site, it will undergo a power stroke that will cause it to put the actin into the midline, and also will release the ADP and the inorganic phosphate, and also it will transfer it from high energy to low energy state. Then we need ATP. ATP is needed here to detach the actin from the myosin. So ATP will act to remove them from each other, and also after that the ATP will be hydrolyzed. It will be broken down into ADP and inorganic phosphate, and the energy that's released from the breaking down of the ATP will transfer the myosin from the low state to high energy state, and the cycle will continue. So we have the binding of actin and myosin, then the power stroke, release of inorganic phosphate and ATP. Then we have ATP will detach from each other, and then ATP will be hydrolyzed, and we will have again the same circulation. I would like to mention that during the contraction, why does not the thin filaments, I mean, for example, if we are pushing them forward, can they go back in the opposite way? So what's really happening that there half amount of the myosin head, they are going to pull at the same direction, while the other half of the myosin heads are going to stabilize this structure. So that is preventing from the going the opposite side. Can one answer? Okay, that's no problem. Can you see? Okay, what's that? Yeah.
In case of rigor mortis, after a few hours, skeletal muscles and the whole body will become stiff. What's the background of that mechanism? The myosin heads will not detach from much.
That's correct. So the question was, most of you didn't see the question, so I will just read it. In case of rigor mortis, after a few hours, skeletal muscles in the whole body will become stiff. What's the cause or the background of that mechanism? That the myosin heads will not detach from actin. I will just explain it right now. So in rigor mortis, yeah. After death, the muscles in the body, they will become stiff. And that usually happens after 3 to 4 hours after death. So what's the physiological cause of that? So as I said right now, ATP and calcium, they both play an important role in the contraction of the muscles. So after death, ATP will not be produced anymore. So we have a little amount of ATP that's stored in our body. It will be consumed in 3 to 4 hours. And then the rigor mortis starts. How does it start? So the calcium will be released, and there will be no ATP to restore it in the sarcoplasmic reticulum. For that reason, there will be a state of continuous contraction, and there will be no ATP to detach the myosin and actin from each other. For that reason, there will be a continuous stiffness. And it usually peaks at 12 hours, and after 2 to 3 days of the death, the protein itself will break down and will degenerate. For that reason, there will be this condition will disappear. Right. Yeah, that's another question. During cross-bridge cycling of sarcomeres, which of the following steps requires energy in the form of ATP? Wait. Adham? Yes, so my part ended here. However, the slides, we have a problem with the laptop, so the slides are not updated, so we will just try to fix it. Yeah, yeah, we can answer.
The second one.
The second one, all right.
Okay, so now let's continue. Have you ever asked yourself how the same muscle can have different power and duration of contraction? Like my hand, I can use it with only fine movements to write with a pen, but I can also, for example, slap a volleyball with my maximum force, for example. So how do we have this ability of changing the force and also the duration of the contraction? Like holding a box for 15 minutes or just holding my laptop for 2 minutes and then I leave it on a table or something. So to understand this concept, we first need to know how does the body deal with controlling the muscles. So we said at the beginning that each muscle has at least one nerve. But is it realistic to control my whole, for example, biceps with only one nerve? Like I contract all the muscle fibers on the in the biceps, or I don't contract all of them? And it's not logic, right? You need more than one neuron, each one with a specific portion of the muscle to adjust the strength and even the direction sometimes of the movement. Okay, so we need to understand this important concept which is called motor unit. So what is a motor unit? Does anyone want to? Okay, okay.
One nerve and the muscle fibers connects.
Okay, exactly. So as you can see here, a motor, exactly as you said, it consists of one neuron and all the muscle fibers it's innervating. So a single nerve or a single neuron won't only also innervate one muscle fiber because we need a lot of nerves at this rate. So a single neuron can innervate more than one muscle fiber. So what is a motor unit? It is like the whole package. This neuron, the purple one for example, and these two muscle fibers which are being innervated by this one. So because of this classification, because of this distribution of neurons on the muscle fibers, it enables us to control exactly the power of the muscle. If we need just very fine movement, we are going to activate only motor unit 1, which is going to act only on these muscle fibers, for example. But if you want a stronger contraction, we are going to stimulate all the neurons, as you are going to see in the different types of stimulation. So all of you got the motor unit idea because the base of the difference between the contraction. Okay, so a single neuron can innervate only as few as 4 myofibers or as high as several hundreds. So it depends on the muscle, as we are going to see now. So some terminology before we continue. We need to know what is muscle tension and what is load, or what is the difference between them. So muscle tension is the force your muscle is exerting on the object. Like when I'm trying to pick up my laptop, I'm exerting force, right? This force is the muscle tension. While the laptop itself, which is the object, it's also exerting force on my muscle, which is due to its weight or gravity, right? It's like resistant. It is resistance to my movement, right? Which is called the load. So we need this when we describe the different types of contraction. Okay, so what is a muscle twitch? A muscle twitch is like the reaction or what was going to happen in the muscle due to a single stimulus, which is not realistic because we are not stimulating the muscle for a single day. We are not stimulating the muscle for one time and then we are leaving it, especially if we need longer duration of contraction. But this one they made in the lab to experiment on the muscle and understand more what is going through the muscle during the contraction. So it consists of 3 different parts. Each muscle twitch is consisting of 3 muscle parts. And as we said, muscle twitch is the what is going to happen in this muscle or the reaction of this muscle to a single muscle stimulus. Okay. So at the beginning we will have a latent period. So when the signal reaches the muscle, it will take a few milliseconds for the previous mechanism that Waleed explained to happen, which is the action potential to travel and all of the gated channels to open and the difference in the ion to happen. So it will take a few milliseconds, which is the latent period. So you won't see any change in tension, in the muscle tension. Yani, there is no power output from the muscle in the first few milliseconds, okay? And then the period of contraction, which is the building of the tension. As we said, the tension is the force your muscle is exerting. So it builds up, of course, gradually in a period of time after it receives the stimulus. And of course, the muscle won't stay contracted forever. It will relax after this. And the period of relaxation is usually longer than the period of contraction due to the difference in speed in the channels that causes each process. Okay. So of course, the contraction period is when the cross bridges are happening. The latent period, the excitation contraction coupling, which was just mentioned, is still occurring. That's why there's latent period. And the period of relaxation is calcium being pumped back into the sarcoplasmic reticulum, which is causing the muscle to relax gradually. So here is just a comparison of different muscles to show that some muscles contract very fast, but also they relax very fast, while others take longer time to reach the full contraction, but also takes much longer time to reach relaxation. Okay, so it's different according to the muscles and the main job of this muscle. Okay, so here it's also showing the individual twitches again. What is happening? We have a stimulus, we have a latent period, and then the contraction is happening. It hits the peak, you don't need contraction anymore. Or it's just a single stimulus reaction. It will take time to relax. And then another one, okay? So, of course, a single contraction or a single stimulus won't keep the muscle contracted for longer times as we may need them in real life scenarios. So how can we keep the muscle contracted and how can we increase the strength of this contraction? First of all, here if you realize in the graph, it's not very smooth, but in real life, when you move your muscles or do any activity, it is very smooth and you don't feel like the muscle twitches, you don't feel each single muscle twitch happening, right? So what is the secret behind this? It is the graded muscle contraction, which is step by step. It's increasing the contraction step by step. So we can increase the contraction step by step using 2 different ways. First one is increasing the frequency of the stimulus. As we said, we have a motor unit, which is a neuron innervating a group of muscle fibers. So this neuron will keep sending stimulus again and again, with less period of time between the stimulus. So the first one took 5 milliseconds, and then it will take 4 milliseconds, and it will keep sending stimulus in an increasing rate to increase the contraction, okay, or the tension of the muscle. And of course, don't forget that you have a refractory period. Refractory period is the repolarization. You can't override that. Like you can't, if you're in the refractory period, if there is any stimulus to the muscle, it won't react to it. So as you can see, we still have partial relaxation. Like it's not from the peak of the contraction you build on it. No, you have to give chance to the muscles to partially repolarize, and then the other one will start midway from the relaxation, which will cause a higher peak this time. Okay, which is increasing in frequency. It's called temporal summation because it's depending on time, as you can tell, because it's saying or sending the same stimulus again and again in an increasing frequency. Okay. So at the end, after we've reached like the power we need or the contraction we need, we have something called unfused patterns. This, you have a question? Okay. This is used if you want to hold something in position for a long period of time. So you need your muscle to stay contracted at the same intensity, no problem, because you're already holding the object, but you need it to stay contracted. So of course it can't be straight line. Why can't it be straight line? Can anyone tell? Okay.
It has to repolarize somehow.
Exactly, because we have refractory period, don't forget that we can't override it. So it will be very small relaxation period, but the overall or the average tension is enough to get the job done. So it's here called unfused tetanus because they are like unfused, they are not connected, there are small gaps in between or small relaxation period. Okay? It's also called incomplete tetanus, as you can see, because we have another time now. Okay, so as you said, there are two different mechanisms that can achieve the graded muscle contraction to make it smooth and in a longer period. The first one was the temporal summation, while the other one is the recruitment. So we said this, like motor unit 1, which is a single neuron with its muscle fibers, the neuron itself is increasing the stimulus. So it's stimulating the same muscle fibers again and again, which will increase the tension, as we just mentioned. So what if this motor unit isn't enough? Because we have many motor units in the same muscles. What if you need a stronger contraction to hold heavier object? You are going to increase the strength of the stimulation, which will increase the motor units involved. We had motor unit 1 which was purple and motor unit 2 which was red. Instead of only using the purple one for fine movement, no, you need a much stronger contraction. So you will use both of them at the same time. So you need to increase the strength of the stimulus, so both of them sense it and work in the same time, which will cause more muscle fibers to be like affected and contract. Okay? So yes, multiple motor unit summation. It's also called like this, and it's from its name specifically to increase the motor units involved. Okay? And this one is mainly for increasing the strength. You don't actually realize this mechanism until you try to hold a box which is empty, for example. But you think that it's full. So I see a closed box. I assume it's full of books or papers, for example. So I try to hold it, and then suddenly it goes flying. Why? Because I assumed it's heavy. So from the beginning, I increased my brain increased the strength of the stimulation, which caused a lot of motor neuron to get activated. But actually, it was much lighter than this, so I didn't need all of this power. So suddenly you feel this like feeling or experience. I think you can relate to that. So here also we have what's called fused tetanus. This is only achievable in labs when they are experimenting on muscles, because the physiology or how the muscle works prevent this from happening in real life in your body. Because as you said, this will damage the muscle. And also, why can't you override the refractory period? Exactly, to protect the muscle and prevent fatigue as much as possible. So here, fused tetanus, it's only achievable in labs. And your body can do this. We'll try to prevent this as much as possible. So here we have a question. Which of the following events best describes the latent period of muscle twitch? Okay. Which one? The first one? So cross bridges are active from the onset to the peak of tension development. So, okay, what do you think? Two cross bridges begin to cycle, but muscle tension is not yet measured. So yeah, it's 2. Because latent period, it is the few milliseconds in the beginning where the muscle is receiving the signal, but still it didn't have the effect of the muscle. And as he just explained, we have the action potential, it's moving in the T tubule, and then it will open the calcium channels, right? So this takes a few milliseconds for the calcium to reach the troponin to start the contraction. That's why there is a latent period. So yes, cross bridges is beginning to form, but there is still no measurable tension happening. Okay, so it takes time. Okay, which of the following features is not related to a graded contraction obtained by a temporal summation? Okay. Okay, repeating a stimulus without changing its intensity, which is not related to a graded. Sorry. So okay, does anyone have any other?
C.
Exactly, C. Aslan, repeating a stimulus without changing its intensity is temporal acceleration. It is just repeating the same stimulus again and again, so it will have a bigger effect. But yeah, see, increasing the frequency of stimulus can override the reflection period. We said no. Okay, so we have three types of stimulus according to their effect on the muscle. We have sub-threshold stimulus. Do you know what is the threshold?
Is the minimum known that it's measured?
Okay, so in order for a muscle contraction to happen, the voltage has to reach the threshold, the depolarization. Like maybe you have many depolarizations, but it's not enough to reach the threshold. I think it was plus 30, I'm not sure, I'll check the book.
That's good.
Plus 30, yeah? Okay, so you need to reach a voltage of plus 30 for a contraction to happen. If you reach, for example, plus 20 only or positive 20, there, nothing will happen. So what do we call a stimuli that will make the voltage reach only 20? A sub-threshold stimuli. So it is a stimulus which will not cause any contraction. A threshold stimulus is a stimulus which it is the first stimulus that is enough to cause a contraction, which will make it exactly 30. And the maximal stimulus is the stimulus which will enable the muscle to reach its maximum capacity. It will activate all of the muscle of the motor unit, and it will have the strongest effect. And we all have limits to our muscular power, right? So the stimulus that is needed for your muscle to exert its full power is called maximal stimulus. Okay, so the last two concepts here for my part is the size principle. So the recruitment process is not random. We said that they recruit different muscle fibers according to the need, right? Maybe only motor unit 1 or maybe 1 and 2. So it is not random. On what does it depend on? Like how does the brain identify I'm going to use only these muscle fibers for now, not the other ones? Okay, so
For example, if you're going to look at an object, you can see the size. If it's smaller in size, our brain is going to interpret it using like less force.
okay, so it is by using the size principle, you are correct. But how is this mechanism? How does this mechanism work? It happens in an ascending order. Yani for two reasons. First of all, to preserve energy. Second of all, to make the contraction smooth. Like our muscle movements are smooth, right? They are not like robotic movement. So what is the reason behind this? Because the smaller motor units are activated first. So it's a gradual or a graded muscle contraction, as we just mentioned. So it happens in an ascending order from the smallest to the largest. And the smallest motor neurons have the highest excitable neurons, because we said motor units is two parts, muscle fiber and neuron. So the neuron of the smallest motor units is the highest excitable, to get activated first. Do you understand this concept? While the largest motor units have less excitable neurons. What is the reason behind this? A small stimulus only activates the smaller motor unit, while a larger one activates the larger less excitable motor units. So why is this principle important? I think we just, okay, your answer. Why do you think it's important?
Because to control the force we apply to raise different.
Okay,
So you don't exert unnecessary force. So for example, you don't want to use more force for something that's like less.
yes, exactly.
You don't expend unnecessary energy.
Okay, so mainly to control the contraction power, as we said, and also to preserve energy if you don't need the. And it's not only about preserving energy. It won't feel natural and it won't feel comfortable if you are just moving your muscles more than needed, especially when you're doing a fine movement like writing, for example. Okay, so here is a size principle in a graph. You start with the smaller one. You needed a stronger contraction. You go to the medium-sized one and then to the largest one. You can also, a very small example, maybe like a fight. The smaller brother is highly irritable. Anyone in the street can make him feel angry. That's why he's highly excitable, but he is weak. So you will get his bigger brother or older brother, which is a little bit more wise, so he's not as like easy to get to frustrate, but he's still in medium power and medium patience, you can say. And we have the father with the most wisdom, so he's not easily. It will not be easy to make him angry, but he has the strongest force. You can remember it this way. It's already easy. The size principle. So, what is a muscle tone? Okay.
Partial contraction to maintain stability or
Exactly, to maintain stability and posture. So right now, I'm standing, my leg muscles, my lower back muscles, or not only the neck muscles, but most of my muscles, all of them are in a muscle tone state, which is a very small contraction that is always there for two things. First of all, to make the posture, and preserve the posture and stabilize the joints. Second of all, to make the muscle ready for any sudden movement. It's like a car that is already ready. You know, you need to heat the car. Especially all the cars, you need 2 to 3 minutes for the oil to move in the engine and this stuff. So this muscle tone is the exact same principle. It makes the muscle always ready for contractions, sudden contraction and powerful contraction. Okay? Yes, exactly. This is the importance of muscle tone. We have two types of contractions, isotonic versus isometric. So, of course, iso means like stable or the same. So, tonic and metric, what do you think?
Tonic changing the length, metric does not change the length.
Okay, so iso means is stable, so tonic is changing in length, yes, but metric it comes from meters, maybe if you want to remember it, meters, which is the length. So here the length is stable, here tonic is the tension, so the tension is stable. We're going to see it now. Isotonic contraction, as we said, okay, so as we said, isotonic contraction, the tension, which is the power or the force the muscle is exerting, is constant, but the muscle length is changing. So, can you give me an example in our real life? When is it isotonic contraction? Okay.
When you're lifting up your bag or something.
Okay, exactly. So when you are moving objects. So for example, this laptop is 2 kg, so I need a certain power to lift it. And now I don't need to increase this power anymore. I'm just moving it around. So my muscle will keep this tension, which is needed to lift this 2 kgs, for example, constant. While the length will be changing, no problem. Like for example, I'm just moving it around, so the muscle is lengthening and decreasing in length. And actually, we have a name for each process when it increases in length and decreases in length, called concentric, which is the muscle shortens, and eccentric, which is the muscle length or increasing in length. So you can remember it, concentric, it comes from concentration, so the muscle fibers are in like a less area or less space, and eccentric is the opposite, it's easy. So both of them are isotonic contraction, where the tension is constant because you are able to move the object you want, but you're just moving it around, so you need to change the length to be freely movable. Isometric is the opposite, and also from the name you can identify it, the length is not changing. So when are you going to, but the tension is changing, when are you going to have a contraction where your muscle length isn't increasing or decreasing, changing?
When the load exceeds the tension, the load.
Exactly, that's exactly the thing. So when you are trying to move an object, for example, I'm trying to push the wall, so my length, my muscle length won't be changing. Why? Because I'm not moving the object. So my muscle is not moving as well, right? So exactly, when the load, which is the force, the object is exerting on your muscle is much higher or higher than the muscle tension, it is an isometric contraction because the length is not changing, but the tension is changing. So at the beginning, I'm pushing slightly, I can't move it, I'm increasing, but you can't see change in the length of my muscle, but actually I'm trying more and more until I can't, of course, it's a wall. So usually trying to move objects that you can, where the load is greater than tension you just mentioned. Okay. So here a very important note that cross bridges are still forming, although there is no changing in length or sliding. So they are still forming, but they can't produce the enough force to make the whole sliding happen. Exactly, because you can't move the object. So during an eccentric isotonic contraction, which sarcomere component maintains a constant length? Okay. Amen? Okay, so first of all, eccentric, we said increasing in length, so just revising it very fast. But yes, as we said before, this is from the previous part, A band never changes in length because it is determined by the myosin part for zero خلاص, and it doesn't change, it doesn't move. Sorry. Or change it. So now I'm done with my part. I hope you understood my part clear. Do you have any questions?
I think the threshold.
Sorry?
The threshold. That's minus 55 and skeletal muscle.
Sorry, minus 50. The voltage?
The threshold.
I'm sure it was positive. Okay, you don't have to memorize it anyway, so it won't affect your study. But I will check it and. You just have to know the definitions, not the exact value. So now Waleed will continue with you. I hope you understood everything. And you have any questions, please do not
So again, hello everyone. So I will continue now with 9.6, providing energy and ATP. So ATP is very important in muscle contraction. It's the main source of energy in the muscle contraction, and it has many functions, including the first one, the move and detach of the cross bridges. It's also responsible for bonding calcium back into sarcoplasmic reticulum, and also activation of sodium-potassium ATPase bond that's responsible for pumping sodium out and potassium into the cell after excitation into contraction coupling. However, the problem is that the amount of ATP that's stored in the muscle cell is limited and it's few, and it's consumed in the first 4 to 6 seconds. So how we are going to continue contracting and moving? So the solution is to provide energy and reproduce ATP by one of these 3 ways. Okay, anyone answer this question? Which of the following is the correct mechanism by creatine phosphate reducing energy for muscle contraction? Direct phosphorylation of ATP, direct phosphorylation of ADP, direct dephosphorylation of ADP, or direct phosphorylation of A and B? Yeah? B. B, that's right. Okay, so for creatine phosphate or direct phosphorylation pathway, simply it's the reaction between ADP, the precursor of ATP, and the creatine phosphate. Creatine phosphate is high molecular energy. It's stored in large amount in the muscle cell. Compared to ATP, it's stored 2 to 3 times more than ATP. So we have large amount of creatine phosphate. It will react with ADP in a process that's catalyzed by creatine kinase and enzyme, producing ATP and creatine. So it will transfer its phosphate group from creatine phosphate to ADP, producing ATP and creatine. This process is very important to provide about 15 seconds of energy. It produces 1 ATP per 1 creatine phosphate and also produce creatine. So I would like to mention that creatine phosphate reserve, or the amount that was already stored in the muscle of the creatine phosphate, we are going to use it during contraction. So before contraction again, or during the state of relaxing, we should reproduce this amount. Okay, for anaerobic pathway and aerobic pathway, they both start with the glycolysis. Or does anyone know, have any idea about glycolysis? Yeah.
It's basically the breakdown of
Of glucose. Glucose. So glucose, either from glycogen breakdown in the liver or from the bloodstream, from the food you eat and from the bloodstream, it will be broken down in the cytosol or in the cytoplasm into pyruvic acid. And this pyruvic acid destination will depend on the situation. In anaerobic pathway, we don't have oxygen. It's anaerobic. For that reason, it's going to be converted in the cytosol or the cytoplasm into lactic acid, and which will be released into the blood, and it will only produce 2 ATP molecules. So comparing it to the aerobic pathway, it produces only or a small amount of ATP comparing it. However, it will happen in much faster rate. For example, comparing it to the aerobic respiration, it happens at 2.1 faster times than the aerobic pathway. To provide energy for about 30 to 40 seconds or slightly more of exercise. So both the amount of the ATP that already stored and also with the creatine phosphate and the anaerobic pathway, it's to provide energy about for 1 minute or of extreme exercise. For the aerobic pathway, it's also starts with glycolysis, a breakdown of glucose, pyruvic acid. However, we have here oxygen. So the pyruvic acid is going to enter the mitochondria. It will be broken down totally into CO2 and H2O and also produce 32 amounts of molecule of ATP. So comparing it to the anaerobic pathway, which produce only 2 ATP molecules, here we have 32 molecules. So to produce large amount of energy. However, it's coming or producing ATP in slower rate. So it's slow but produce large amounts. Okay, so to provide energy for hours of for example, if you are working for hours, that you are depending on Arabic pathway. Other substances that we can use other than pyruvic acid include amino acid and fatty acids. So fatty acids are usually consumed, start to be consumed after 30 minutes. When you already consumed all your ATP, creatine phosphate, or the glucose or glycogen that you already stored, when all of that is consumed around 30 minutes, then you are going to start with fatty acid breakdown into the aerobic pathway. Okay, for the muscle fatigue, it's a state of physical inability to contract, even though the muscle is continuously stimulated. So we are stimulating the muscle, we are telling it contract, contract, contract. It's not contracting anymore since it's inability. There are 4 mechanisms that are involved in muscle fatigue. The first one is ionic imbalances, as I said. For example, sodium and potassium, they play an important role in the whole contraction topic. So if there's continuous contraction, after some time there will be ionic imbalance, so we need some rest, time to rest and to restore this balance. The second one is increased in organic phosphates. Inorganic phosphates can disrupt or interfere with the release of calcium from sarcoplasmic reticulum, and also will interfere with the cross bridges of the myosin. The decreased ATP and decreased magnesium, low ATP and decreased magnesium also will interfere with those processes, and also we have decreased glycogen stores. I would like to mention that lactic acid and pH, some people they are saying that, for example, muscle fatigue is a result of lactic acid and pH. However, the truth is that lactic acid and pH, they only contribute to the sensation of pain. They have nothing to do with muscle fatigue. So lactic acid and pH, they are just making you feel pain, not causing anything with muscle fatigue. So we have 2 types of fatigue patterns depending on the exercise that you are doing. For example, if the exercise is short and intense exercise, for example, if you just sprint for 5 seconds, you are going to feel fatigue fastly, and you will not be able to continue running. However, you are going to recover quickly. On the opposite side, if you, for example, long and low intensity exercise, for example, such as running a marathon, you will continue for long hours and you will not feel fatigue fastly. However, the recovery will be longer and you would take hours to days to recover from this long exercise. Okay, so for EBOC or EBOC, it's okay. So during contraction, we consumed all the ATP that we have, creatine phosphate. Also, we consumed it. Also, we consumed the glycogen. We broke down into glucose, and then glucose will be consumed. And also, we converted pyruvic acid into lactic acid. So all of that, after finishing the contraction or the exercise, we need to restore all of that. So we should restore reproduce the ATP, the creatine phosphate, the glycogen, and also the oxygen that we consume. And also we reconvert lactic acid into pyruvic acid. All of those reactions require excess amount of oxygen that be consumed. For that reason, this physiological state is called as excess post-exercise oxygen consumption, or also called oxygen debt. So you are going to use the oxygen in large amount during anaerobic respiration and also during the rest state, and then you should use it. Regain this oxygen that you already used. Okay, for 9.7, the force, velocity, and duration of skeletal muscle contraction. I will start with the force of contraction. So force of contraction, it depends on the number of the cross bridges that are formed, which is in turn is affected by 4 factors. The first one is the number of the muscle fibers that are recruited or contracting at the same time, the size of the muscle fibers, the frequency of stimulation, and the sarcomere stretch level. So if we have large amount of muscle fibers that are recruited, so they will act together, they will produce more forcible contraction. Larger and bulkier muscle fibers will also produce more contractive force. Increase also stimuli will cause the same thing. However, for the sarcomere stretch, I would like to show you this diagram. As you can see here, the optimal or the perfect sarcomere operating length is near to its resting length. In this length, there's the ideal overlapping of the myosin and actin together. So if there's extremely stretching the sarcomere, the myosin and the actin will be stretched away from each other. The myosin will not find anywhere to attach to. As you can see here, there's no connection between them. There's no overlapping. For that reason, they cannot just pull them. There's no connection between them. For that reason, there will be no any contraction here. However, on the opposite side, if it's the sarcomere is greatly shortened, there will be an excess overlapping between them, and the actin filaments would interact with interfere with each other, and this will reduce the amount of force that's produced. So the optimal or the perfect sarcomere stretch is near of its resting level. the velocity and duration of muscle contraction, it explains how fast a muscle can contract and how long it can continue before it stops. It's influenced by 3 factors: the muscle fiber type, the load, and recruitment. For the muscle fiber type, we are going to classify the muscle fibers into 3 types depending on 2 factors. The first factor is the speed of contraction, depending on the speed of the myosin cross bridge forming. So we are going to divide them into slow and fast fibers. For the second point, we are going to talk about their way of producing ATP. So it can be oxidative or glycolytic. The oxidative, they are going to use which type of pathway? Aerobic, while the glycolytic will depend on the anaerobic pathway. So depending on those 2 factors, we are going to classify them into 3 types: the slow oxidative, the fast oxidative, and fast glycolytic. Slow oxidative, as explained itself, it's slow contraction, slow cross bridges forms, and also depends on aerobic pathway. While the fast glycolytic on the opposite side is fast contraction, fast myosin ATPase activity, and also anaerobic glycolysis. So fast oxidative is somewhere in the intermediate between those 2 types of fibers. I would like to mention that it's very important to memorize this table, everything in this table. I'm sure that you will see it in the exam. So please try to memorize everything in this table. Okay, for the load and recruitment. Load refers to the force that the muscle refers to the force that must be moved or overcome by the muscle contraction. It affects the speed and the length of contraction. For example, if you are contracting or moving without any load or light or no load, the muscle contract faster and will stay for longer time. However, if the heavier load, it will cause longer latent period, the period before the contraction starts. It will reduce the shortening time, and also it will cause slower shortening. So it will cause slower contraction and small duration of contraction. For the recruitment, it's the effect of motor neuron recruitment. The more motor neurons that they activate more muscle fibers will produce faster, stronger, and longer contraction. Okay. The muscle of the back that help in maintaining our posture will show which of the following features: few mitochondria, fast fatigability, aerobic synthesis of ATP, or large glycolysis of ATP. So, as Karim said, aerobic synthesis of ATP. For exercise and muscles, so muscles change in response to how much they are used. So if they are continuously used and in large amount, they become larger, stronger, and more efficient. If they are not used, they will become weaker and smaller due to atrophy. They undergo different changes depending on the type of exercise that they are going through. For the first type, we have aerobic or endurance exercise, such as walking, running, or even sprinting. This type of exercise causes physiological changes in skeletal muscles, including increased number of capillaries surrounding each muscle fiber, increased number of mitochondria inside muscle fibers, and increased myoglobin synthesis. Can anyone explain what the mechanism behind each one? What's the source of each one? For example, why we need a higher number of capillaries surrounding each muscle fiber? Yeah.
For nutrients.
Yeah, for more oxygen to be provided and more nutrients also. For the number of mitochondria, more ATP production. Yeah, right. For myoglobin, more oxygen. This type of exercise is important for increasing the endurance and also fatigue resistance. It affects all types of cells of muscle fibers, mainly the slow oxidative, since they depend on the aerobic pathway. And continuous endurance or aerobic exercise can cause the conversion of the fast glycolytic into fast oxidative fibers, increasing the aerobic capacity. So for the second type, which is resistance or anaerobic exercise, examples such as weightlifting or isometric exercise, such as my colleague explained. For example, if you are applying too much force while the muscle is not shortening, that's called isometric exercise. Main effects of skeletal muscle, it causes muscle hypertrophy. Hypertrophy is resulted mostly from one, increase the size of individual muscle fibers. So the number of the muscle fibers, it's the same. However, each muscle fiber itself is going to increase in the size. Increase mitochondria, myofilaments, and myofibrils. Increase glycogen stores and also connective tissue between the muscles. So all of those causes will lead to hypertrophy or the increase in the size of the muscle. So those adaptations can convert fast oxidative fibers to fast glycolytic. and require only a few minutes of exercise every other day for continuous gains. Okay. So as I said right now, so muscle cells, they depend on the exercise that they are going through. If they are not used, they are going into a type of atrophy. It's called disused atrophy. The strength of the muscle can decline at the rate of 5% per day if it's not used. For example, there is no muscle tone, nothing. There is no any type of stimulation, and over time it can be atrophied to 1/4 of its initial size. And the fibrous connective tissue is going to replace the lost muscle. So we are going to lose muscle fibers, and it's going to be replaced by fibrous connective tissue. And complete recovery will be hard and will take longer time than the period of immobilization. For example, if you stop moving, or for example, if anyone got in an accident and stopped moving their leg, for example, 2 months, gaining back the amount of muscle that they lost would take 4 months, for example. So the amount of time that will be take to regain the recovery will take longer time than the immobilization period. Yeah, can anyone answer? Yeah, when I said for career, that's a wrong question. I remember this. That's the one that's strictly one. So, no.
Fast fatigue.
Yeah, see. So this question was really tricky, and many of us got it wrong this week. So here, aerobic synthesis of ATP in bodybuilder. So depending on the resistance or exercise, there is anaerobic. So it's not totally wrong. Few mitochondria. I just mentioned that the resistance exercise increase the mitochondria amount. Mitochondria, as you can see here, glycogen also will be increased. So the only possible answer is that there will be fast fatigue. Okay. That's it. Thank you everyone.